What is the state of the intelligent design movement? How has the conversation about origins shifted in our culture and in the church? Well, we've got the perfect guest today because he's not only a Biola professor, Doug Axe, but has also written a book on intelligent design called Undeniable.
What's at stake is your posture toward science.
Can we just make peace with Darwin and move on?
Is there something particularly and profoundly important about humanity with respect to the rest of life? A lot of people who are believers aren't happy with this call to make peace with Darwinism because they say there's something huge at stake here. We cannot just brush this off and carry on as Darwinists. Why would a Christian feel as though they need to make concessions to what I consider to be one of the weakest, most pathetic scientific theories that has ever come out in the history of science? I'm on record saying that.
To our third session in the Human by Design series. Tonight is going to be, I think, a lot of fun. I hope a lot of fun. We're going to be talking about kind of closing the loop on irreducible complexity and defining intelligent design, what that really is, how we can make the argument from design from a logical and philosophical standpoint. And we're going to talk a lot about information. How important is information? How do we use it? How do we get it? And how do we know that that information comes from an intelligent source? So here's our roadmap. We're going to look at why things are not an accident, why things that work, if they work, they're pretty much not an accident. How do we build things from scratch? What does that process look like? We'll talk about, very briefly, because I think it'll be a pretty slam dunk case, but why chemical evolution doesn't work. Chemistry doesn't lead to information. Then we'll look at complexifying the soup, the primordial soup. How do we get complex stuff out of primordial soup? Okay, a short course on how it works, and that'll actually be. We'll spend a fair amount of time on the information, how we do get complex information out of the molecules that build life, and then we'll wrap up tonight with the theory of biological design. This is a new way of looking at how life originated, and it's a really cool concept. I think you'll like that. And I've renamed it from what the original guy did. He calls it continuous environmental tracking. I call it pre-programmed limited adaptability. We'll talk about why that's a more simple, more inclusive way to characterize what he's talking about. But I think it's a really refreshing way for us to characterize what we believe and why. So, as you know, I've been introducing my dad into the story, and he's got a connection this week as well. You know that we found his diaries from World War II, and this was my dad as an airman, as a radio man on C-47s. He was a rather dashing young fellow. That's actually, he was probably about 27 there. The picture in the upper left, he was probably 32 when he got married. In his diary, one of the things that we found several pages in was his entry for D-Day. And this D-Day entry is fairly straightforward. It just says, D-Day. Dropped paratroops without incident on Cherbourg Peninsula, France. These were the troops who later took Saint-Mary-Glace. One of the hugely important battles of the D-Day invasion was this city called Saint-Mary-Glace. We'd been there. We went there when we did our Normandy trip this year. It was a kind of a lifelong dream to do that. So how did they get the troops there? It required a lot of information, and that's our connection tonight. When they did the D-Day invasion, and I'm focusing mainly on the troop carriers, but this is a map of where the troop carrier, basically the cargo airplanes went. There were 821 cargo airplane missions involved with D-Day. That's a lot of airplanes in a fairly small amount of airspace. And one of the places that they took off for on this map is Kosmor, which is where my dad was based. These guys that flew across the channel, my dad was carrying paratroops, but they did two different kinds of mission. One was paratroopers being dropped out of the airplane, and one was this towing of gliders. Extremely dangerous work. You can see there's a cable going back to the glider, and they're fairly close formation. You'll also notice in this picture, this is from D-Day, that the fields are flooded. The Germans flooded the fields in Holland in northern France, so that it would be difficult to land. And so, of course, that makes it really difficult for the paratroopers. There were paratroopers who landed in 3 feet of water with so much equipment they drowned in 3 feet of water. It was nighttime, especially with the overnight drops, and my dad's mission was at midnight drop. The gliders also were looking they had moonlight, but there was a lot of cloud cover as well, so a lot of the gliders had trouble with these flooded fields too. Very treacherous, obviously, in a lot of different respects. So you don't get all this stuff to work without a lot of planning and foresight and putting it together. But just think of some of the things that were involved. On the 6th of June, 1944, there were 160,000 troops that landed in Normandy. 73,000 American, 83,000 British and Canadian combined. Over 10,000 casualties, which includes killed and wounded, around 23,000 airborne troops, and again, 4,100 aircrew members flying those 800 missions. Airborne divisions, there were 13,000 paratroopers, about 4,000 infantrymen, glider infantry, so they were going in on the gliders. Very complex mission. It was the most complex military operation in history. They had issues with communications, with weather. There was a big deception where they tried to convince the Germans they were going to attack someplace else. You had logistics planning, attrition. You had to have enough equipment that when stuff broke or was lost, you'd have backups. You had land, naval, and air forces. You had 3 principal nations coordinating and others as well. You had to resupply. You had to have the resupply coming over before you even finished the landing. You had medical support because there were going to be injuries, and you had 15,000 bomber missions, over 15 times the number of cargo missions. So now when I flew years later, it was a little bit different. I flew this kind of airplane, and we had all kinds of information. We had a radar down here someplace. We had a head-up display, enemy warning systems, all kinds of stuff that they didn't have back then. But they somehow made it happen. Now, as far as complex as all that was, however, it pales in comparison to what's happening in your body just as you're sitting here. It's an amazing thought, but that's really what we're focusing on tonight, is the information complexity of D-Day was nothing compared to what is happening in you right now. You are fearfully and wonderfully made. And that's from a Bible verse that we'll look at, but first we have to look at my granddaughter. The fact that this baby comes from a fertilized egg in 9 months. She's 6 months old now. But there's far more complexity there than there was in D-Day. There are verses that talk about that. Psalm 139 is one of our more common ones, but it says, "You created my inmost being. You knit me together in my mother's womb. I praise you because I'm fearfully and wonderfully made. Your works are wonderful. I know that full well. My frame was not hidden from you when I was made in the secret place. When I was woven together in the depths of the earth, your eyes saw my unformed body." God planned all that out. That's the only reasonable explanation we can come up with. So let's look at why chemical evolution won't work. We talked about before, one of the ideas is the chemicals just came together by accident. We looked at that in the previous week. So in this short video clip, listen for the feasibility of naturalistic processes leading to life. Okay, that's your assignment in this section.
2 questions. How do higher forms of life evolve from lower forms of life? Charles Darwin answered that question, or at least thought he did. But the other question is one that Darwin never even attempted to answer. Where does life come from in the first place? Scientist and philosopher Stephen Meyer and chemist James Tour. Uncommon Knowledge, now. Welcome to Uncommon Knowledge. I'm Peter Robinson. We're filming today in Fiesole, Italy. Stephen Meyer earned his undergraduate degree at Whitworth College and his doctorate in the history of science at Cambridge. Now the director of the Center for Science and Culture at the Discovery Institute. Dr. Meyer's books include Signature in the Cell, Darwin's Doubt, and The Return of the God Hypothesis. James Tour received his undergraduate degree at Syracuse University and his doctorate in synthetic organic and organic metallic, I can't even say what this field is, in synthetic organic and organometallic chemistry from Purdue. Now a professor at Rice University, Dr. Tour's areas of research include nanoelectronics, graphene electronics, silicon oxide electronics, and carbon research into what the Rice website terms, quote, environmentally friendly oil and gas extraction, close quote. Dr. Tour has published more than 1,000 peer-reviewed research papers and holds several hundred patents. Steve and Jim, benvenuto. Darwin. A couple of quotations here. As I mentioned earlier, there are two pieces to Charles Darwin. And the second piece is evolution, his elaborate theory that random mutation and natural selection can lead to, or did lead to, the creation or origin of many different species. Now, that we won't go into it, agree with it or disagree with it. It's an explanation, it's a body of thought, it's a kind of discipline. But the other piece, the first piece, is this. Darwin simply assumes that life exists. His theory calls for evolving from something that's there in the first place. Here he writes in a letter to a friend in 1871, if, and oh what a big if, we could conceive in some warm little pond that a protein compound was chemically formed, ready to undergo still more complex changes. That's Darwin in 1871, more than 2 decades after he published On the Origin of Species, and he's still wondering how life might have originated. Now Richard Dawkins, contemporary Oxford zoologist, quote, Darwin made it possible to be an intellectually fulfilled atheist. But of course, Darwin only made it possible to be an intellectually fulfilled atheist if you believe, this is a question, but it's my surmise, you can only say that Darwin made it possible to be an intellectually fulfilled atheist if you believe that that prior question, where does life come from in the first place, is either trivially easy to answer or in some basic way doesn't matter. Is that correct?
Or it has been solved, but neither of those three conditions have obtained. It's not trivially easy to solve. It hasn't been solved, and it is actually quite an important part of the whole naturalistic story that Darwin and modern neo-Darwinists have attempted to tell. And you're right, there's a distinction between what's called chemical evolutionary theory, which attempts to explain the origin of the first life from simpler non-living chemicals,
right,
and biological evolutionary theory, which attempts to explain the origin of new forms of life from simpler pre-existing forms of life. Darwin's theory was a form of biological evolution, an attempt to explain the origin of new forms from simpler forms, but he presupposed, as he put it in the origin, one or very few simple forms at the base of his famous tree of life, and did not account for where that came from simpler prebiotic chemicals.
Before we continue, and I've got loads more to ask, obviously, why am I, a layman, coming to this a century and a half after Darwin publishes On the Origin of Species, scratching my head and saying, fellows, this first question, where does life come from in the first place, that's a pretty big one. How is it that a sort of interested layman can only suddenly realize, you guys had this elaborate, what about this? Did science simply treat it as unimportant or so easy that they'd get to it sooner or later? The question of the origin of life.
Nobody knows the origin of life. Nobody has ever known the origin of life, but people assume, very much like the Babylonians assumed, that it came out of a stinking pond. That's where they say their life came out of, and their gods came out of that same pond. And that's the primordial soup model, and that model persists today. You look in any textbook from elementary school to advanced college textbooks, and it will speak of the primordial soup model. Life came out of that there were molecules that came together and they formed higher order structures, and that formed a cell, and that led to life. That is a bunch of nonsense.
So he doesn't really pull his punches there. Chemical evolution is a bunch of nonsense. Okay. So what do you think about the feasibility of naturalistic processes leading to life? Not real good. Okay, I'm not planning to go any further in chemical evolution because it seems intuitively obvious, if you're open to this kind of logic from people who really know what they're talking about, that it is not a possibility. So we're going to move on. Any objections to that? Anybody want to go deeper into it could have accidentally happened? And I don't mean to belittle you if you hold that view, but we'll talk afterwards or whatever. Okay, all right. So how do the other part of the issue they talked about is, so you've got chemical evolution that doesn't work. The other one is biological evolution to make more complex or make novel features that aren't there before, to make those out of what exists. And that's what Darwin focused on. How do we complexify? And I looked this term up. It's actually a real word. I thought I was making up kind of a funny word, but it's actually a mathematical term for some kind of mathematical proof. But how do you make things more complex? We're going to look at a little bit of the complexity that exists when we talk about how, and then we'll talk about how it could have been made more complex. So you've seen this video before. I've watched it countless times and love it, so I'm going to subject you to it again. It's only about 2 minutes on what proteins do. Now, proteins are the molecular structures in your body that do functions principally inside cells. And so this guy is going to talk about some of those cells that are involved in the reproduction process.
So this is one of the biggest molecular structures that you have, at least as far as we've discovered so far, inside of us. So this is a single chromosome, and you have 2 strands of DNA in each chromosome. One is bundled up into one sausage, the other strand is bundled up into the other sausage. These things that look like whiskers that are sticking out from either side are the dynamic scaffolding of the cell. They're called microtubules, but the name's not so important. But what we're going to focus on is this red region. I've labeled it red here, and it's the interface between the dynamic scaffolding and the chromosomes. It is obviously central to the movement of the chromosomes, but we have no idea really as to how it's achieving that movement. We've been studying this thing called the kinetochore for over 100 years with intense study, and we're still just beginning to discover what it's all about. It is made up of about 200 different types of proteins, thousands of proteins in total. It is a signal broadcasting system. It broadcasts through chemical signals, telling the rest of the cell when it's ready, when it feels that everything is aligned and ready to go for the separation of the chromosomes. It is able to couple onto the growing and shrinking microtubules. It's transiently, it's involved with the growing of the microtubules, and it's able to transiently couple onto them. It's also a tension sensing system. It's able to feel when the cell is ready, when the chromosome is correctly positioned. It's turning green here because it feels that everything is just right. And you'll see that there's one little last bit that's still remaining red. And it's walked away down the microtubules. That is the signal broadcasting system sending out the stop signal, and it's walked away. I mean, it's that mechanical. It's molecular clockwork. This is how you work at the molecular scale. So with a little bit of molecular eye candy, we've got kinesins, which are the orange ones. They're little molecular couriers.
So the words that were flashing up on the screen, scrolling up, those are words that I added. That's not in the original video, but you can look for this video online. It's a much longer video, and it's fascinating. So let's look at what some of the other things are, what some of the other processes that proteins accomplish for us. So we looked at this one. It's involved in reproduction. Sometimes proteins typically fold into functional shapes. Some act as valves and pumps, and some are motors that propel bacteria. And they're all doing it at once, automatically, not being supervised. Maybe it annoys you that all these voices are going at once, but all those things are happening in your body simultaneously. That's why the voices are going to make that point. And it's all automated. Think about the idea of building a machine that rebuilds itself. That's what's happening here. This microtubule one, just to emphasize that one, these are not, you would think they'd be strands along which the gene is sliding. but it's actually being destroyed or decomposed or taken apart on one side and put together on the other so that it propels it along. And it does that in real time every time your cell reproduces, every time a cell divides. This is happening to all the genes. It just boggles the mind. Incredibly complex structure. So how do we code for all this stuff? Well, it takes information. So this is a animation of a DNA, portion of a DNA molecule. And you've seen this structure before, where you have a sugar backbone, and these colored areas in the middle are actually the nucleobases. Okay? Now, in order to reproduce, the first thing that has to happen is you have to unwind this chromosome, which is always twisted up. And once it's unwound, then it's all more parallel, and the chromosome can be divided into two separate sections. And there are proteins that do all of these functions. This is animating what's happening, but it's not showing you how it's happening. So if we focus on one of these lines, and these are all color-coded, they're not color-coded in real life, just so you know. But we have 4 different basic nucleobases. There's adenine, thymine, guanine, and cytosine. I am not a geneticist, so this is all layperson's description of the process from an outsider. So if any doctors in here that can help me, if I make a mistake, feel free to. Oh, good, I'm off the hook. No, but seriously, this is just research I've done to get the basic big picture. I'm kind of a big picture guy. So if you look at these things, they're always paired up. So on the opposite sides, you may have noticed there were different colors. So adenine and thymine are always paired up. Guanine and cytosine are always paired up on opposite sides. And some different diagrams, like there's a later diagram where they're colored differently, so there's no consistency on how people color them for illustration purposes, but they're always paired up on the opposite sides. So if there's an error every once in a while, that's a mutation where one of these nucleobases gets changed. Okay, so that's a single bit of information that gets changed. We'll talk a little bit more about how that happens and how there's actually an error correction method that the cell uses to find these errors and to fix them. That'll be our long story short video at the end, is how we fix this stuff. So if we look at this individual set of information, a very short piece of it, we've got these letters, and we've got codons that are among the nucleobases. So every 3 of these nucleobases is paired into a codon. ATC, this one is a stop. There's several stop codons. And anytime the molecule is being processed to build a protein, and you get to one of those stop signals, it breaks there, and that becomes the design for the protein that it's being coded for in that section. So in this section, you've got 3 codons. You've got one that's, we're just labeling A, and then one B and one stop codon. So there's 3 codons in this set of 9 nucleobases. Everybody with me so far? Okay. All right. So here's what happens when we build when we try and reproduce, again, simple terms. So this is a chromosome. It's got a whole bunch of those nucleobases, and they look something like this. And you can see that the A and the T are opposite each other, the G and the C are opposite each other. If we're going to make a new, if we're going to cell divide, we have to duplicate this. And so the first thing that we have to plan on is replication. So we're going to split up the 2 sides of that. So we've got this part that's up in the upper left now is split up into 2 separate strands, okay? And we want to replicate it so that we end up with 2 of these things down here, okay? The process works like this. It's called transcription. And in order to get transcribed, we pull one of these pieces over here, and the transcription process changes all of the A's to U's, and then we get the T's to the A's over here, G's to C's, and so on. So we have uracil in place of, so there's no, any place there was an A over here, it's the uracil over here, okay? And then we have a translation process. When we end up, we started with DNA, which is deoxyribonucleic acid. We end up with ribonucleic acid, and that's what we need for reproduction. We also have a thing called translation, where this happens down here. Oops, let me go back for a second. This happens down here. This is messenger RNA. That's what the mRNA is. And then over here, once we have our new code built for RNA, we end up with a mechanism that separates and grabs one of these codons. This one attaches to the next codon, and this is the amino acid stage right here. Those are the amino acids that actually build the proteins. Okay? And so we have this amino acid attachment site and anticodons. I have no idea what they are, but I found a cool picture, so there you go. So we have these different amino acids, and here are 4 of them, for example. We've got the 4 down here, and the same color codes are over here, so I just labeled those with a box. And then here's the table of all the possible combinations of nucleobases. Okay? I know we're not going to stay here very long. Just bear with me for a second. So you have basically 4 different things here. You've got U, C, A, and G. Up here you have U, C, A, and G, and over here you have U, C, A, and G. And these are the 4 different amino acids that were coded in the previous slide. So to decode this thing, you look down the row, across the row, and then across the row. So you get the U here, the U here, and the U here, and we get UUU. And we can go to the next line down. It'll do the same thing with UUC and so on. So you get 64 possible combinations. You get 4 times 4 times 4, 16. 4 times 4 is 16, times 4 is 64. Okay. So there's 64 possible combinations, but you only end up with 20 amino acids because several of these can code for the same amino acid. I have no idea how that works or how the cell knows that those are amino acids that function the same even though they came from a different codon set. So when you look at all this, then you end up with 3 of these that can be coded with 6 different codon sets or different nucleobases that make these amino acids. And then you have the 3 of them that are stop codon sets. And then if you count up all the others, you end up with, wait for it, 20. Okay? So they're the 20 amino acids, the stop set, and so you get 20 possible amino acids out of 64 possible combinations. What I don't know, and I don't know how important it is, but I don't know how important it is whether these different kinds of codon sets that make this amino acid function differently in different settings. There's so much to this stuff. It's amazing. So the steps in review are this. You get nucleobases, the 4 different kinds, the A, G, T, and C. Then you have codons, which are combinations of 3 nucleobases. Messenger RNA. You have exons but no introns. We'll get to that in just a second. And you have proteins. The simplest protein in the body is 44 codons. The most complex, the longest one is 38,000 plus codons. Really long. Has to be. So if you're going to do a spelling bee, you don't want to get that word. Okay? That'd be a really tough one to spell. Okay, so this is our picture we started with before. There's another really cool thing about these deals. Not only do you get 9 amino acids making 3 codons, but it turns out if you jog one over, you can get an entirely different set of codons. So it can be grouped such that you have these 3 making codon C, these 3 making codon D, and so on. But wait, there's more. If you jog one over again, you can get a 3rd level of information out of a single strand. This is like taking the book of Genesis and going 1 letter to the right and starting pairing up things like that, and you get the book of Matthew. And 1 more book over and you get the book of Revelation. It's embedded codes within embedded codes. This is like the ultimate puzzle. But wait, there's more. Because it turns out you can go backwards. And you go backwards again. And you go backwards again. It's possible theoretically to get 6 different codes out of the same piece of DNA. Talk about an efficient designer. Can you imagine the complexity? It just boggles the mind. Now, to be fair, they've only found places where there are 4 overlapping codes. They've never found any with 6, but they theoretically could exist. But even then, think of 4 different books coming out of the book of Genesis, just by jogging over 1 character. It just boggles the mind. So they've discovered that there's different ways that overlapping can happen. You can have overlapping, non-overlapping, comma-less. This one's non-overlapping with a comma. I guess that's if they skip one. And then you use all these different combinations to make the codons do what they're supposed to do. And this is all, then there's different, you can go different directions, you go parallel, nested anti-parallel, all this stuff. And it's all controlled with what's called the gene regulatory network, which we used to think junk DNA was junk, because that's what the evolutionists told us. It turns out that much of the gene regulatory network is involved in regulating which codons come out of which sections. It's like the operating system. And they keep finding more and more stuff in the non-coding regions that they have been calling junk DNA, which is no longer thought of that way. But you'll still see it in the textbooks and online. You'll see junk DNA all the time, but there is no junk DNA. They haven't found the function for all of it, but they found functions for a whole lot of it. So back here, there's our error. And we're going to get back to that in a second. But a simple protein, again, we talked about a simple protein. It typically has like 150 molecules, 150 codons, which means 3 times that number, 450 base pairs. One functional protein by accident. According to Doug Axe, who was in that first video clip saying that evolution was a pathetic theory, he's done the math. He did it at Cambridge. And he's come up with the logical mathematical proof that it's 1 in 10 to the 74th power that you could get a protein that functions by accident. Now, it might not be the right functional protein, but it would be a functional protein that does something, okay? Stuff in the cell is being broken down and recombined all the time. It doesn't have a single purpose. Like a given protein that's involved in metabolism or reproduction isn't static. It gets broken down, repurposed, recombined into the protein du jour, whatever is needed at the moment. So this stuff is happening constantly. So you need the right ones. But that, by the way, this number is more than the particles in the Milky Way galaxy. So that's not atoms, that's like the quarks, you know. So it's just an amazingly big number. In humans, we have about 50,000 functional protein types that they've identified. And they keep coming up with more all the time. The number may be bigger by the time I go home tonight. Of those, what Doug Axe also found out was that 5 to 10% of the 50,000 are unique to each species, which means that if you have a chimpanzee or you have a lizard or any mammal, take us and badgers, 5 to 10% of the 50,000 proteins, which is 2,500 to 5,000 proteins, are unique. They're not found anywhere else in nature. Now, the 80% might well be found someplace, or 85% might well be found someplace in nature, but they can't they can't say that all of this was co-opted from another animal and transmogrified into another species, because they're unique. So we've got that one functional protein, 1 times 10 to the 74th power, likelihood of it coming about, but we've got between 2,500 and 5,000 of them that we need all at once when this organism starts. So it's pretty much game over for any evolutionary biological accident to cause a new feature. So we're familiar with instruction sets, and that's what DNA is. But we look at simple things like a Lego set. So when we build a Lego set, we have a set of instructions, and we're used to following instructions, and if we do get something out of order, we end up with something at the end that doesn't look like the picture on the box. And then you're in trouble on Christmas morning, right? So we have to follow the instructions, and DNA has a set of instructions with this gene regulatory network as well. We're familiar with instructions in all kinds of examples in life, not just Lego sets, but cooking recipes, climbing stairs, map directions. They all require a precise, ordered operation. Protein length, again, we talked about the difference in lengths a little bit. Titin is a good name. Crucial for elasticity in muscle tissue, that's the 38,000 codon protein. The shortest protein, about 44 amino acids, takes 44 codons and 132 nucleobases, and there are about 400 codons in the average protein. Okay, so let's look at how we would get an accidental combining of information such that we get a new protein. So here's a strand of protein, colors are different from before, but it's the same idea. So if we just make a single strand we're going to look at, there's only a few things that can happen with mutation. You can get a break, and if you have a break in the protein, it's possible now you have a gap. And if you have some random other piece of protein floating around, it's possible that you could get that to join into the existing set of broken proteins. And if they happen to connect, you've got a new set of information. That's a mutation. You can also have a loss of information. That piece could come back out. And you could end up rejoining the original. So you can have a break, you can have an addition, you can have gains or losses. What you can't get is new functional information big enough to describe a new protein. That's 450 nucleobases. So it's really difficult to think about how you could get a new feature out of accidental change. So if we need 44 codons, that's 132 nucleobases, okay? So here's my diagram with 132 nucleobases. That's a lot of individual bits of information that have to be in the right order. So if we take a look at this, there's one other feature I mentioned earlier, introns and exons. Now we're not talking about gasoline, but these are terms I hadn't heard before. Exons are nucleotide sequences that carry information for protein synthesis, and they are to be exported, whereas introns are interrupting sequences that do not code for any proteins but are found between exons. So it's not like we just have this set of information that's straight through. Because introns are removed, this term in our HN RNA is heterogeneous nuclear RNA. And you have the basic gene with this mix of information. Some of it's usable, some of it's not. But it has this mixed together. So that means we need a whole lot more than 132 in a typical DNA strand. It looks maybe more like this. And then if we take and we color code a bunch of information that's not going to be in the eventual DNA, that's going to code for a protein, and then we recombine it so that we have only the ones that are going to code, 132 up here, we have a whole bunch that's also got to be filtered out. And that's also part of the gene regulatory network. So I was obviously bored, so I figured out, do I have the right number here? And it turned out I was too short. See, I've got a couple dangling nucleobase here. So what do? I just grabbed two others, but when I counted these up, I found out I only had 43 and I needed 44. Okay, so what do? I just grab two random ones and shove them in there, and that gives me, like magic, number 44. You can do that with animation. How do you do it with a cell? Okay. It's just, again, it boggles the mind. Okay, so we're going to do a little exercise. So everybody should have some notes. And on that note, on the back of your front page, you have these words. Okay? Now, you can cheat. You can talk to your neighbor. You can do it individually, but you got 2 minutes. Okay? So the challenge is make a sentence with as many of these words as possible in 2 minutes. Ready, set, go. No pressure. Okay, time is up. So I know there's at least one correct solution. Did anybody, other than Kit, and don't tell us what your solution is yet, but did anybody use all the words other than Kit? Good, okay, so we had some pretty good success there. Now there's a number, you guys are smart people, there's a number of ways you could have attacked this. One is you could just put all the 2-letter words, 3-letter words, 4-letter, 5-letter, 6-letter words together, okay? Another way you could have attacked it is to say alphabetize it. That would have taken quite a bit of time, unless you do it with magic. Or you could start building phrases. The cat is around the mouse, or the cat is around a mouse, okay? And start building ideas like that. But one of the things you didn't have to solve was, are the words upside down? Are they right side up? Now, if I'd taken a bunch of pieces of paper and thrown them in the air and put them on the ground, some of them would have been upside down. Some of them would have been upside down, and so on. Blank page, words on the other side of the page. So there's a lot of things that this oversimplifies. You could also have, you could start again getting a sentence like this. So a couple of you that that we're successful, read me your sentence. Somebody, what'd you get, John?
The cat is chasing the mouse around the tree at the end of the street today.
Okay, that's good. There's a couple of places you could have put today, but you made a logical sentence that's grammatically correct. That's good. You could have made a shorter sentence with a few words left out. You could have done this one, the mouse is at the end of the street today chasing the cat around the tree. I put today in a different place. But it makes more logical sense because you have some logic and some knowledge of cats and mice and what these symbols actually mean to put those together. The amino acids and the codons don't have any grammatical structure. They don't have an intuitive ability to place themselves next to the right adjective, adverb, etc. So the problem is just compounded every way you look at the nucleobase generating new information problem. It just gets worse the more you look at it. So here's a short, just 1 minute summary of how this process works one more time.
Let's have a quick reminder of how all of the stuff in your genome works. The basic idea is that your genome contains information in the form of DNA. This blueprint comes in the form of the nucleotides, the A's, G's, T's, and C's, as well as the precise order in which they are arranged. TTA or CCG, for example. When it comes time for your cell to make a new protein, your DNA unzips, and a copy of the code made of a molecule called messenger RNA, or mRNA, is made. It uses a molecule called uracil, U instead of T, but it uses the same genetic code as DNA. This mRNA is then transported to a protein factory called a ribosome. There, the ribosome reads the open-faced mRNA in 3-letter chunks called codons. Each of these codons corresponds to one of the building blocks of proteins called amino acids. As the ribosome reads the codons, a molecule called a transfer RNA, or tRNA, brings in the matching amino acid. The ribosome brings everybody together and links the amino acids together in a chain in the same order as the codons in the mRNA. This goes on until the ribosome hits a signal in the mRNA to stop, at which point it releases the amino acid chain, which is now ready to become a finished protein. And in this way, our cells can turn just 20 amino acids into pretty much all of the stuff we are made.
Okay, I think he's about to take a breath, but that's kind of the process. Again, it's really complex. I like this guy because he really shortened it, but as soon as I cut him off, right after this, he was going to go into how there's junk DNA and it's all degraded and all this stuff, so I cut him off, sorry. So we've talked in previous weeks about irreducible complexity. If you reduce the complexity at all, you don't catch less mice, you catch no mice. And this week we added to that a different kind of mousetrap and talked a little bit more about the proteins. The inference to the best explanation is every time we see something that has highly ordered, refined, functional parts or information in any kind of logical pattern, all of those things always come from what? What's the source of those things in every instance? Some kind of intelligent agent. It's never accidental, random chance. So we're going to talk next about this idea of the theory of biological design. What I want you to listen for is what are the problems with using the language of materialism when we use their language to talk about a process that we really think was not naturalistic or accidental, but was intelligently guided by God. What are the problems when we use their language to describe what God did, okay? So that's what you're looking for in this next section.
Really important what we're going to be talking about today in terms of replacing Darwin's sacred imposter. And in fact, what we're going to be really discussing in terms of all of these things are, man, I went back to this, is mutation selection, our self-destructive compromise. And if I were to ask you, and I'm not even taking a raise of hands here, how many of you believe that evolutionary thinking tends towards atheism? I bet you I would get the vast majority of your hands there. But if I were to ask you, go out to a friend and talk to a friend and explain to them, tell them, how does evolutionary thinking tend towards atheism? And go through the details of how it takes a person who might believe in God and take them over to atheistic thinking, you would probably struggle with that. So, all Christians can take their witness of Christ as the Creator and Savior to the next level by learning 3 enlightening truths. Three enlightening truths for all of us. The first is this: that creatures and human designed things share corresponding engineered features or engineered parts. This is something new, and we haven't been really teaching this for a very long time, but it's absolutely essential that we learn this. Second, that believing in adaptation, and I'm speaking specifically today about adaptation, by random mutations and natural selection is anti-theistic because it is anti-design. And number three, the mutation selection explanation harms both science and theology. So these are the main topics that we're going to be covering today, and I think if we get these down, then we'll be able to enhance our witness with others around us. So first.
So this is Dr. Randy Galuza. He is the new president, and well, a couple of years ago, Institute for Creation Research, which is only, Jason tells me, 17 miles away in Dallas. It's an excellent museum. They have talks over there, and they focus on honoring God as creator. And it's just an outstanding place. We'll talk a little bit more, but everything in the next section of this briefing is from Randy's talk. I heard him give this talk a little over a year ago, and it really was groundbreaking for me. It changed the way I think about some of these things. So what are the problems when we use their language? Again, I'm just for emphasis. The three problems he cited are that first of all, we need to understand creatures and human designs share corresponding engineering features. That's really important. We know what engineering features are. These chairs, this TV, my computer, they're all engineered, they're designed, and we as biological entities share engineering features that we've just talked about. Information coding that is similar to engineering features that we see in everything around us, that we can identify those things that are engineered. Second, believing that adaptation happens by random mutations and natural selection is anti-theistic because it's anti-design. You can make design and God co-equal, not co-equal. God is not less, design is not less than God. God is much more than design, but design has to be part of God's nature. So if we're using language that ascribes what God did to chance and accident, we are demeaning the idea of what God did and not holding what he did in awe as we should be. And third, the mutation selection explanation harms science and theology. It's a disservice to science because it undermines what actually happened with the engineering process. And it's demeaning to theology because it's inaccurate and, again, falsely characterizes what God did. So the takeaway is that our language that we use shows our worldview. It shows the lens. We talked about this last year when we did the worldview series. We look at the world through a lens, and our lens has to include the creator and sustainer of life. It has to include the fact that he has a plan for us through Jesus Christ to come to a relationship with him. That's all part of the plan, but it includes biological design. So when we use this naturalistic language, it conveys that we didn't need God to get to where we got. Some of the scriptures that talk about this: the heavens declare the glory of God, the skies proclaim the work of his hands. Psalm 8, 3 through 6. I won't read all of this, but some of the key ideas, it's the work of his hands that we're seeing. The work of your fingers, you have set it in place. The work of your hands. The psalmist did not use compromised language, and we don't need to do that either. A couple of other verses from Psalms and from Romans. So, in the beginning, you laid the foundations of the earth, and the heavens are the work, again, of your hands. This consistent message, the psalmist uses very high language to describe what God did. Yeah.
Can I say questions to the end?
Oh, you can do questions anytime you want. No.
I'm having a hard time understanding what those 3 points mean in our witness.
Okay.
Like, we can't use the word mutation when we're talking to somebody else. I'm not sure.
Yeah, so if the idea is if we ascribe changes in species to accidents, for example, to accidental mutation changes, what we're really saying is we didn't need the designer to go from badgers to cats to dogs. But in fact, what the DNA portion of this talk should have helped you understand is that all of those changes are complex. They can't happen by accidental mutation. We can talk about mutation, which does happen. We know that bacteria does have mutations, but it doesn't ever increase in complexity. Does that make sense?
That part makes sense. I'm still trying to understand what point you're trying to get to about don't use their language and what those 3 points actually mean.
Right. So let me go back to those points. Let me just go back here.
Don't get hung up on.
No, that's okay. If you have a question, somebody else probably does as well, but I appreciate your boldness in asking. So there's another short segment where he's going to talk about the language. Let me just do that. We'll come back to that because I think I'll answer your question a little more effectively after he's done his next little bit. Anyway, this section out of Romans 1, God's invisible qualities, his eternal power and divine nature have been clearly seen, being understood from what has been made, so that men are without excuse. The point is that we want to honor God with our language. And again, we will come back to that a little bit more. We're going to do a little game show, okay? So here's our game show. And we're going to talk about what Dr. Galuza characterizes as the theory of biological design. It's still a work in progress. We're trying to figure out how we can characterize what God did in the design process in a way that honors him and accurately describes what he has done. So if we're to think about design characteristics, what an engineer would do, what you would do if you're going to design something, what are some of the words that come to mind? Any thoughts? Purpose, okay. Efficiency. Function. Okay, so here are some that he came up with. Purposeful, good job. Regulated, complex, targeted, orderly, specified, fitting and suitable, and precise. These are terms that describe accurately what an engineer does, and when we look at DNA or any design of a biological entity, these characteristics are in play. So when we look at the world of human design, we might see something like this, an automobile engine. And we would see all of these characteristics displayed in this engine. We would also see design characteristics that include things like what it's supposed to do, numerous interconnected parts, particular arrangement, proper alignment, moving parts, precise timing, exact dimensions and shape, fitted for a purpose. So these are all terms that we would use about design when we know it's been designed. And we've been misled, I was misled certainly growing up, to believe that biological features were accidental. And so there's a different set of terms that we would use for that. And so what Randy's going to say is we need to craft a new language. And part of that language is recognition of these characteristics that we see in nature. First, that creatures tend towards stasis. They tend towards stability. They don't tend toward variability. We don't see any animals with a fifth leg or a tail turning into a leg. We don't see these kind of changes in complexity in nature, or when we see them, they're usually a non-functional entity. He's got a leg that doesn't work. He's missing a leg, but we don't see additional features that are helpful. No creatures that we know of who have sight in the visual spectrum can suddenly see infrared. Not aware of any humans who've developed that characteristic. So if we look at anti-design characteristics, this is where we use the other terminology that's associated with evolution. So some of these terms would be things like purposeless. And what Randy did was he combed the literature, and he only used terms that he found in at least 4 separate research articles that were peer-reviewed. So purposeless regarding DNA, random, accidental, trial and error, broken, unplanned, cobbled together, messy. So back to your question, when we use these kind of terms to describe the DNA that we see in nature, we're using their language to say it happened by accident and that it was a mutation that led to a purposeless, random, accidental trial and error process. And we end up with a bunch of broken DNA that we characterize as junk DNA. When we use that language, we're mislabeling what reality is really composed of. So here are, just to demonstrate what the evolutionists actually say about their own processes, here's just 2 quotes. Randy gives a lot more in his talk, but we're abbreviating his talk a bit. Okay, the core tenet of the modern synthesis, which is Darwinian, neo-Darwinism, is that adaptive evolution is due to natural selection acting on heritable variability that originates through accidental changes in the genetic material. Such mutations are random in the sense that they arise without reference to their advantages or disadvantages. And then the idea when we talk about it this way is that nature naturally selects those which are more functional or have a survival advantage. But in fact, we find that mutations are universally bad. We don't find good mutations that help us to adapt and develop new features. Here's another quote. A classical Darwinian evolutionary system embodies a basic principle, purposeless genetic variation of reproductive individuals united by common descent coupled with natural selection of those rare individuals that fortuitously express the traits. It's a process replete with chance. So when we characterize organisms this way, we're not using the language of a purposeful, intelligent designer who created with intention. And so that's the change that Randy's advocating, that we start thinking about how we use our language. Does that make more sense?
Except for the fact that I don't know any person that understands the flaws in evolution that would ever say that and mean it. So I'm not sure what language is supposed to be changing.
Okay, well, these are the leading thinkers. This is a guy who's written a book, Clonal Evolution and Cancer. And what will happen is they'll write something about biology, but they'll have these kind of throwaway lines in here about this stuff happened through accidental random changes, aren't we lucky? You'll find it in research papers all the time. Most of us don't read research papers all the time. I don't read them all the time, except when somebody like this pulls these quotes out. So you find these assumptions embedded in research all the time, but we're so used to them, we just tend to overlook them. And we just take them in stride. And what he's advocating is, let's not do that. In our research, let's be more purposeful and intentional in the way we characterize these things. So when we talk about anti-design characteristics, he's going to give an example of where there was a display at the Institute for Creation Research that when they went and looked at it in light of what he's thinking about now, they realized they had to change it. And so here's an example of where they changed the description about cave fish. They're doing research on cave fish, and he'll talk a little bit about that. But here's what the description said. He's going to give an example of that and how it should have been changed. So listen in this video for Darwinian assumptions at a Christian creationist museum before they made some changes.
Let's consider a non-illustration. Ooh, I hate to read this. This was ICR's explanation for cave fish, which we had in our original museum. Read what we said. As genetic information is copied and passed on generation after generation, occasionally there are copying mistakes known as mutations. Mutations have been observed to destroy, damage, and corrupt genetic information or to be neutral, but have never been observed to add new information. This is true even of so-called beneficial mutations that may be advantageous to the surviving organism in some creatures. When a mutation occurs in a light environment that causes animals' offspring not to have eyes, it's an enormous disadvantage. So natural selection eliminates this flaw. When the eyeless defect occurs here, that is in a cave, it does not give any disadvantage, so it is not eliminated. In fact, it gives advantages. Those eyes can crash into things, injuring the eyes and get diseases of the eyes, possibly leading to death. Eventually, selective pressures ensure that all are eyeless. These ghostly fish are prime examples of how mutation and natural selection lead to a reduction of functioning systems. These adaptations are no evidence at all for the belief that complexity has arisen by such processes. They only show how information can be lost in a fallen world. How many of you have ever repeated that? How many of you have been saying that? Why not? We taught it. So let's go over and sum up what we just said. We just said that surface-dwelling features gradually morphed into cave-dwelling forms due to random genetic mutations, which led to broken systems. through a purposeless trial and error process of death and survival that resulted in a loss of information that produced depigmentation and blindness, and selection pressure ensured that all are blind. Why are you laughing? Because we just basically repeated what? Darwin's mechanism. All we did was repeat it. We just repeated it. But then we put the caveat, but this shows how you can break something but not make something. Do you see what we did? We jumped in and we played Darwin's game on Darwin's field by Darwin's rules. And that's why we haven't killed this after 50 years of teaching against it, because we basically said everything he said was true. And once you get into that, you can't win. You can't win. And so for years we've basically been saying, well, Darwin's mechanism is true, but it's inconsistent, or it's insufficient, or it's incomplete, or it's inaccurate. But the moment we say his mutation selection mechanism is true, we've lost. For all the reasons I just gave you up to this point. And that's why, by jumping into that, the details, it derails scientific research by not looking for non-random changes. And only once we broke away from it did we start to develop a continuous environmental tracking model, which assumes that organisms are as engineered and much, much, much more than this car, and we're looking for corresponding system elements, believing that they can be explained by engineering principles. We start looking for non-random changes and we start to find them. We had to break away because it was derailing us. Second, it derails scientific search by not looking for the not broken. So when it came to cave fish, the question is, does a broken system cause cave fish depigmentation? Is the fact that they are now these hypopigmented things because the systems which lead to pigmentation, are they all broken in this fish? So we started to look. And we took this blind cave fish and we put it back in river conditions under real sunlight, and in 32 days it looked like this. Same fish in both pictures. Was the pigmentation system broken? Obviously it wasn't. It derailed us. Completely derailed us. Mutations for adaptation, no rational engineer would use it. So no wonder people start to question whether God really created us. Theistic selectionism, it's just the creationists' light version of real evolution. That's why it's bad. It personifies nature as if it's causal, which means that we jump into the same realm as the idolatrous pseudo-creator, the great selector. That's bad for theology. 6, natural selection, we always said it's not a creative process, it's a conservative process. Do you know what that meant when we were saying that? It meant this. God created this to operate after the fall so that some of you, when your children have genetic mutations, they will die before they reach reproductive age, and that saves the rest of this population from getting their genetic mutations. That's what we're saying. That's pretty gross. But that's what we were saying. And number 7, we said that we agreed with Gould's perverse indeed idea. In other words, this, Gould was smart enough to realize that Darwin was after something more than just the diversity of life. He wanted to mock God. And people, when they look at creatures, they see evidences of his goodness, his love, his wisdom, and his power. But what Darwinian thinking says is that everything that you thought was good and that demonstrated the goodness of God really came about through a blind struggle for life and death, where one creature destroys and leads to the extinction of another creature, all for its own selfish advancement. And he says this mocks the Creator God, and he's right. In addition, he has the substitute idolater. So this is why this whole thinking harms. It harms us in many ways.
So that's kind of the summary of the kind of change in language that we need to think about. And that is, as we talk about creatures, as we talk about differences between creatures, we want to do it in a way that uses engineering language rather than accidental language, because it's a mischaracterization. So again, that's kind of my conclusion from this section. But again, these are the same fish that are just 32 days apart. They've also found that fish embryos of these blind cavefish have eye sockets and the seed of what would become an eye, but then sometime during the embryonic stage it gets turned off and the fish is born without fully developed eyes. So what they're doing with this blind cavefish experiment is, over a period of generations, they're exposing generations of blind cavefish without pigment to regular light to see if at some point the eyes will come back. And they're putting these fish in blind or in dark conditions to see if they will lose their pigment and if their eyes will go away at some point. You see, that's where, assuming that the organism has the ability to adapt within certain parameters, leads to looking for features in DNA and in the design of the creature that we would never look for if we didn't think it was designed. We would think it was broken. So, for example, the famous example of Darwin's finches, where you have the different beaks in different shapes that tend to proliferate differently depending on the environmental conditions. There's a certain amount of environmental pressure which will tend to favor certain types of shaped beaks. So in the dry season, you need harder beaks that are able to break into the shells of these seeds that they have to go after. In the more moist seasons, they tend toward a thinner shell beak where they can go after the softer fruits. But they're still finches at the end of the day, and they will go back and forth over generations depending on the conditions. So the idea is that there is pre-programmed adaptability built into organisms, and that those organisms can vary between the limits of what change is possible within that DNA program for that animal. There's limits to how far it can change, but they can change within that limit. They'll never change outside that limit. That's why we can get dogs from Pekingese all the way to wolves out of the same DNA, but they won't ever become cats. They won't ever become badgers, because there are limits to that adaptability. But the environment can, in fact, turn grizzly bears into polar bears. They are the same animal. It's just that polar bears have certain features that have been environmentally pressured into the generations that are living up north versus the grizzly bears. So Michael Behe we talked about Michael Behe with the Darwin's black box idea. He was one of the ones who talked earlier, like 15 years ago, about the limits of it, because the edge of evolution, that's his second book. How far can evolution take us? And it's basically, now we know better through what Randy's talking about, is this limited ability to adapt within a set range. So again, he calls it continuous environmental tracking. I'm trying to get him to use the term pre-programmed, because it is limited, it can't go very far, adaptability, because that describes all the features he's talking about. So that's where the theory of biological design has to include some component of change, because we know that what we used to call microevolution is really a pre-programmed ability of animals to vary a bit. And again, so those are the two terms: continuous environmental tracking and pre-programmed limited adaptability. So in this last segment, we're going to go back to the first opening segment we had, which was with Sean McDowell, who's spoken here before, and Doug Axe, who I'd love to get here, but he hasn't spoken here before. But they're going to talk just a little bit about what's at stake in this idea of science and faith, and what is intelligent design. How is that different from creationism? So those are the two things to listen for in this short clip. Oh, how does ID differ from creationism? Yeah.
Tell us a little bit more about what exactly is at stake in this debate over intelligent design.
Well, in the broader question of what's at stake on the question of are we created or are we cosmic accidents, just that is at stake. Who are we? Where do we come from? Where are we going? Which is huge. But are we asking, like, what's at stake if you take an ID position versus these other positions? I think what's at stake is your posture toward science. And we also haven't mentioned theistic evolution. So that's another one of these positions that's common out there within the Christian community. They have different postures with respect to Scripture and different postures with respect to science. It can be the case, it's not always the case, that young Earth creationists can say, Scripture first. If I encounter something where it looks like science is in tension with Scripture, I chuck the science and I go with Scripture. And old Earth creationism could be that as well. I'm not saying that everyone is offering that, but that can be. Whereas intelligent design is saying, let's just look at the science carefully and see what the science actually says, and not spin it in the direction of materialism.
So one of the distinguishing marks of the intelligent design movement is that it specifically is not trying to do that harmonization of science and Scripture.
Yeah.
So that's just not even on the table.
Yes, but the interesting thing is, so it's saying let's do the science honestly, and when you do the science honestly, it ends up beautifully harmonizing with the big story of Scripture. Okay, and I'm not trying
but not on the details.
There's lots of details that remain to be figured out.
Fair enough.
And I think it's great that people are thinking about that. I also don't think that we have to get answers to some of those details. The big questions are the things that we really need an answer.
So that's kind of his summary of where creationism and intelligent design fit together. So if we're to summarize those things, what's at stake in the discussion of science and faith? We don't want to give up either science or faith. We think they fit together. We think that they build on each other, that they're cohesive. What is intelligent design? How is it different from creationism? Anybody want to take a stab from what you just heard at what the difference is between intelligent design and creationism? Yeah, Laura.
They're not attributing it to a specific creator or aligning it in Scripture. They're just saying we're going to follow the science, and if it goes this way.
Right, going to follow the science whichever way it goes. But it turns out, as he says, it always ends up in favor of our theology. So science is always on God's side, yeah. But intelligent design, you're right, is only going to follow the science and not going to work toward harmonization with Scripture. It turns out it does harmonize. But their focus is trying to reach people who are open to science but not to theology, to at least get them to the point of, well, there had to be a designer. Once you get them to the point of there had to be a designer, now we can talk about who that designer is. And that's a further step. But intelligent design specifically is looking at the science, whereas creationism is looking at the theology, not always as a first step, but certainly as embedded in the process, and finding that, again, the science is in harmony with the idea of a creator. So that's the real difference. Is intelligent design is an intentional effort of the science establishment of typically believing, at least deists. They're not all Christians, people involved in intelligent design. They're at least deists who believe that there is a creator. And again, that's the first step toward theology. Is now once we've established that there is some intelligent designer, and we look at the characteristics. And Stephen Meyer, in his book, Return of the God Hypothesis, does this really well. We've talked about that before when we did the Science and Faith talks. But he says the beginning of the universe shows that all the material functions, all the laws of nature, gravity, light, all those constants, the strong and weak nuclear forces, all those had to come into being and be perfectly balanced at the start of matter, time, space, all of those things. So if time, space, and matter happened at a point, you can't explain time, space, and matter with the materials. You have to explain it by something outside time, space, and matter. So it's timeless, spaceless, and immaterial. It's incredibly complex, as we've looked at. So it's some entity incredibly intelligent, had to be incredibly powerful to generate the volume of the universe that we see, which is incomprehensible. It had to be purposeful, and we know that people make choices, that individuals, persons, we say, make choices. So God, at some point, whatever the entity is, we attribute it to God of the Bible, made a point-in-time decision to use his timeless, spaceless, immaterial power to create intelligently and complex things. And so we end up with a timeless, spaceless, immaterial, purposeful, intelligent, complex, powerful entity. What do you call that? If it's Darwin, you call it chance. If it's Dawkins or Gould, you call it chance. But we don't do that. And so that's the point of trying to use the proper language to describe what God did. Okay, any questions at this point? Any other? Are you a little more comfortable, John, or still? I think what I'm hearing you say is you don't encounter this kind of materialistic language very often. Is that?
Not from people that understand the wholes, evolution
Yeah.
is taught by materialists.
Right. No, you're right. And people that understand this stuff won't use that language. But people that are writing, again, we go to the loudmouths, the Richard Dawkins, the Jay Gould, who's now passed away, Christopher Hitchens, Daniel Dennett. Those guys, in the 90s and early 2000s, they were called the four horsemen of the atheist apocalypse because they were writing all these books. They've stopped because there is no new information coming out on the side of evolution and naturalism. But we still see these voices, we hear these voices in the research establishments that will not cede to God or to any supernatural entity the creative power that we see in the universe. And so that's what we're trying to rebel against, if you will. And again, if you're reading the local newspaper, you're not going to see this language unless you're really looking for it carefully. It is there, but it's an assumption that's kind of underlying everything else. So any other questions? Yeah. Oh, just stretching. Stretching is a good thing. Well, the only thing we've got left is we've got a long story short video on that thing I alluded to earlier, which is how do mistakes, when they occur, how do those get corrected? And it turns out that mutations have another problem if they're going to generate a new feature, and that is they generally get wiped out because they get corrected. Again, some of the amazing stuff that proteins are programmed to do. So if anybody want to take a break, you're welcome to do that. We take 5 minutes and come back, or you could leave at this point and watch the video later, but I'll go ahead and hit play unless you guys want to take a short break. It's about 10 minutes, then we'll be done. Okay.
This world is a dangerous place, and I'm not just talking about spiders and saturated fats. The information in your DNA is under constant threat. From things you would never suspect being dangerous, like sunlight, oxygen, even water, are extremely dangerous for your DNA. And that's not even to mention the actually scary sounding things like ionizing radiation, gamma rays, alkylation, chemical mutagens, and other pathogens. Every day, your DNA experiences 10,000 lost single letters of code, and 10,000 cases of damage by oxidation, and 55,000 single strand breaks, as well as 10 of the extremely bad double strand breaks. All of these happen every day in just one of your cells. Just one! Multiply that by the trillions of cells in your body, and and I can't emphasize this enough, how in the actual world can we still be alive with our cells basically burning to the ground every second of every day? Great question. Good news, bad news situation. Your body is like a library of information that's constantly on fire. Luckily, your library has an army of librarians who work tirelessly to search the library, extinguish the flames, and actually rewrite the missing information with extreme accuracy. As fast as the environment burns down your DNA, your body's defenses rebuild it. Without these incredible systems of damage recognition and repair, people couldn't exist. No life could exist, not even the lowly fungus or bacteria. This ability is essential for life, even the very first living organism. Otherwise, life could have never started. And it's actually unfair to compare our body's repair mechanisms to librarians, because librarians usually have eyes to be able to sense problems and brains to solve them and hands to carry out those plans. Your cells need to be able to recognize and fix these quadrillions of problems autonomously, without a single thought or even any help from the Dewey Decimal System. amazing. But how can a molecule, something with no mind, do something as complex as sensing and repairing? It's easy enough to imagine a molecule doing something simple like bumping into another molecule. That's just chemistry. Easy as a rock rolling downhill. And I can understand how a scientist can sense things or modify things, but how can molecules know how a thing is supposed to be? How can they notice when it isn't that way? And how can they do anything to fix it and not make it worse? Imagine a glass of water detecting something wrong with a novel. It can't. It's just molecules. They can't read. They have no knowledge of what a novel is or what it's supposed to say, much less what to do about it if it could spot an error. It'll just sit there, being water. But inside of you, there are molecules that can detect errors in your DNA and much more. You can't live without several different repair mechanisms. We've only got time to cover one of the many systems, and the simplest one at that. Here's how it works. The simplest DNA repair mechanism of many is intimidatingly called base excision repair. Fixes a single damaged letter of DNA, and it's needed in every known form of life. If you had a damaged strand of DNA, first, one machine or protein enzyme scans down it and recognizes the single damaged letter. It would detach it from the DNA backbone, extracting it like an old, dirty tooth, leaving the functioning letters alone. Next, another machine detects the empty spot and breaks a single strand of the DNA backbone. A third machine then snips the DNA backbone again, removing the segment of backbone that's empty. These two enzymes have the ability to break the DNA backbone at will. Lucky ducky you, they only do this when needed, and at a specific site of damage. How could enzymes like this just pop up at the start of life without first destroying all of the DNA in sight? A question for the ages. A fourth machine then inserts the replacement part, an intact matching letter of DNA, complete with a new backbone segment. Finally, a fifth machine welds the replacement part to the backbone on both sides, completing the restoration. This is all oversimplified baby talk, of course. The actual mechanics of these things is far beyond what we can convey here. But all this madness is happening every day in every single one of these tiny little danger hot dogs, also known as bacteria. If your reaction to all of this isn't, "What?" then you're just not paying attention. This is literal submicroscopic surgery with the added monkey wrench of the cell does not stop operating while it's being repaired. Imagine trying to repair a Jeep while it's still running. And remember, base excision repair is just one of the many DNA repair pathways, and not even the most complicated one. Even the simplest organisms absolutely need repair mechanisms. The first life and all life needs repair at the very beginning. In bacteria, the instructions to make these 5 enzymes are coded in DNA, requiring more than 8,000 letters of information. If you're smart, you'd notice, just like the scientist Manfred Eigen, that this is a huge paradox. That much DNA could have never accumulated unless it was first protected by repair mechanisms. But the repair mechanisms themselves require at least this much DNA. This sort of thing is kind of like taking kids hiking in nature. You could probably bring a few of them without losing any. But if you had to bring 8,000 kids on a hike, things get a bit more dicey. No, Joseph, don't touch that! Frozen! Without some system to track them and keep them from wandering away, guaranteed to lose some of them. And so it is with life. Without a repair mechanism, only a small handful of rungs of DNA could be accumulated before information started getting irreparably lost and a death spiral began. But some people say life started simpler. RNA stored information long before DNA existed. Don't you know that? This would only make the problem worse. Because RNA is so fragile and so easily damaged compared to DNA. And we know that life requires many RNA repair mechanisms that we haven't even touched upon, so that doesn't work. And on top of that, there's even more problems that can pop up in your cells outside of DNA that need to be fixed also. Remember, the cell is more than just a library. It's also a factory. And sometimes its manufacturing machines break or produce defective merchandise. So cells need to first identify an undesired or broken molecule, and then they can either fix it, recycle it for parts, or transport the junk to an ejection hatch in the cell for disposal. Here's just one example. The central manufacturing machine inside of you is called the ribosome. It takes the instructions in your DNA and RNA and follows them to build nanomachines. It's the factory or assembly line of the cell. And each cell can have anywhere from around 10,000 to many millions of ribosomes building things all at once. To read the instructions in RNA, it is threaded through the ribosome like a train through a tunnel. But sometimes the process goes wrong. The RNA train can get stuck inside the ribosome tunnel before the thing it's building is finished. A bit like constipation.
Deadly constipation.
And there's no laxative on a prebiotic Earth that can fix this problem. And it turns out it's a quite common problem in all life. Here's just one of your cells. Your body has trillions of them just like this. And as you grow, it'll need to replicate itself. Ribosomes to the rescue. That's just the sort of thing it does. But ribosomal constipation is so common that before this cell could make a single copy of itself, every one of its tens of thousands of ribosomes will get clogged up 5 times over. So life requires a way to detect and fix this kind of thing as well. Floating around inside of the cell of all living things are machines that spot errors and label bad molecules, demolition machines, and recycling machines, among others. Fun fact, 1 in 20 machines in your body are actually for destroying the other 19. That's comforting. This machine can recognize when a ribosome is stuck, so it sticks a label on the half-finished protein called a degron. Then the unfinished protein needs to be chopped up for parts. Luckily, your body has little molecular chainsaws that love to chop up proteins. But hang on a second. Isn't it a bit dangerous to have protein chainsaws floating around inside of you? What if they chop up some useful thing? Wouldn't you know it, they're programmed to only chop up things labeled with a degron. Proteins that destroy other proteins are essential for life. Don't worry, you're safe.
For now.
And then, the defective RNA has to be chopped up too, since if it's let free, it'll just keep clogging up other ribosomes. So a different label is applied to the mRNA and different chainsaws jump into action. Hang on, why not just use the same machines? That seems sort of wasteful. Good question. Sort of like recycling paper is a different process than recycling aluminum, different molecular machines are needed to recycle different biological parts. Maybe so, but some people will object that life started simpler. Without all of this fancy stuff. Okay, ribosomes are admittedly fancy. And yet, as fancy as they are, they sometimes goof and make a mistake. If we're going to imagine that earlier life had something simpler than a ribosome, something more crude, less fancy, it would only be more prone to make errors and produce more junk. This would make the problem worse and not better. The need to recognize and repair things would only increase the simpler life got. Life could never have started without sophisticated recognition, recycling, and repair mechanisms. If they weren't sophisticated at the start, they would have caused more damage than good. They have to detect exactly what needs to be fixed and repair only those molecules. But obtaining these sophisticated molecular machines would require more information than can possibly be accumulated or maintained in the absence of those very repair mechanisms. DNA was designed with the expectation that errors would happen, as well as the foresight to be able to be fixed. Blind forces like Darwinian evolution have no expectations. They have no foresight. Only intelligent beings do. Thanks for watching.
So it's a good thing that we have some repair mechanisms because we couldn't have gotten off first base, right? So pretty cool stuff. So, again, what we talked about tonight, just to summarize, things that are functional are not by accident. Chemical evolution won't work. It's a really challenging thing to build DNA that has functional information, and we need to be thinking about, be observing our language as we talk about how things came into being, and use language that honors God. So where are we in the big picture? We're halfway through with our trek through design pointing to God. We have done the heavy lifting. This was kind of a heavy lifting week. DNA is hard. Next week we're going to look at animals that are really unique. I think it's going to be probably one of my favorite weeks that we cover. Most of our time actually will be in a single movie that is copyrighted, so I can't put it into the clip at the end. All the stuff that I use is coming off of YouTube sources or places where they encourage proliferation. But the one that we're going to look at next week, I'm not going to tell you what it is. You'll have to come to find out, but you will not be able to watch it from the video link that I send out later. I will tell you where you can find it, but Gail and I watched it again the other night. It's one that we purchased a while back. It's just phenomenal. So you will come away in awe of what God has done. But we'll first start with some stuff that we will be able to share online, and that's just all the incredible designs in nature that have no parallel in other animals. So we'll talk about some unique features of animals that just can't be explained by co-opting a feature into another animal. So that's where we are for tonight. Anybody have any questions before we quit? If not, I'll stand by afterwards. You can accost me at will. But let me pray for us and thank God for his incredible work. Lord, we thank you for being the creative designer that is talked about in the Psalms and throughout the scripture in Romans, where the world and your works point to you as the creator just from the things that you've made. We thank you that we can trust you to make good designs that are functional, efficient, beautiful, and that honor you. And we want to honor you as we think about what you have made and as we stand in awe of you because of what we see in your works. Thank you for your word. Thank you for this time tonight, and we praise you in Jesus' name. Amen. Thank you, guys.